The broadcast is now starting. All attendees are in listen-only mode.
Hi, my name is Deirdre Mason, and I am with the Association of State Drinking Water Administrators, and I want to welcome you all to our webinar today with USGS on understanding chemical and microbial contaminants in drinking water, raw, treated, and tap water. So just a little bit about ASDWA. For those of you who don't know, our mission is to protect public health and the economy through the provision of safe drinking water. And our members are the Drinking Water Program Administrators from the 50 states, territories, Navajo Nation, and DC, who provide public water system oversight in the form of regulation, technical assistance, and funding, and also coordinate with multiple partners. This is the third webinar in our series with USGS. Our two previous webinars on April 24th and July 2nd were recorded and are available on our website, and this one will be too. We'll also have two more webinars through the rest of 2018 that you see here down at the bottom, so stay tuned for those. And for today's webinar, our presenters are going to provide information on the spectrum of chemical and microbial contaminants to address key issues and questions of drinking water quality in three segments: one on groundwater sources, one on water quality from source through treatment, and one on water quality from distribution to the consumer. And we have a number of presenters today from USGS that you see here. We have John Sullivan from the town of Billerica, and we also have Susan Glassmeyer from EPA's Office of Research and Development, but not necessarily in that order. So after the webinar, we will have a video recording on our website along with the other two I mentioned, and in the future we'll have the others. We also have a number of other recordings, webinar recordings on our website if you're interested in additional topics. And so with that, I'm going to go ahead and turn the presentation over here to Bruce Lindsey with USGS. Bruce?
Good afternoon. Are you seeing my screen?
Yes, that's great. Thank you.
Okay. Thank you. My name is Bruce Lindsey, and today I will be giving the first of three presentations on water quality. The topic of my presentation is on the quality of untreated groundwater from principal aquifers of the United States. The National Water Quality Assessment Project, or the NAQUA project, has several objectives. One of those objectives is to answer the following question: what is the quality of groundwater used as a source of public supply in the United States? We can answer that question using the following approach. We do spatially weighted sampling of the principal aquifers of the United States, and that allows us to identify what are the contaminants of concern. It also allows us to identify whether those contaminants are widespread across the country or regional in nature. In order to conduct a national assessment, we selected 18 principal aquifers for the study. Out of the more than 60 principal aquifers in the nation, these 18 represent 85% of the groundwater used as a source of public supply. During the time period of 2012 to the present, we have sampled more than 1,300 wells in these 18 principal aquifers. You may note that we have no samples shown in the Central Valley of California and the California coastal basins aquifers, and that is because the California Gamma Project has addressed those aquifers and assessed them using the same methodology as we are using in the NAQUA project. Now, we selected these aquifers based on pumping volume, but we evaluate the aquifers by spatially weighted sampling. That allows us to assess the proportion of the aquifer that has high concentration. For example, using this method, given a grid of 60 cells, we can tell that a contaminant is unlikely to be missed in our sampling if it is present in more than 4% of the aquifer. And most of our aquifers were evaluated with more than 60 cells. We realize that groundwater is a three-dimensional resource. We have other networks that sample the shallowest part of the groundwater using monitoring wells, the slightly deeper part of the aquifer that is used for domestic supply. But we will not be addressing results from those networks today. The results we are discussing today come from the zone of aquifers used as a source of public supply. This depth zone varies by aquifer, but it is typically deeper than those other zones. This zone of the aquifers of the United States represents more than 80% of the groundwater that is used for human consumption, and thus is the focus of our study. In order to evaluate the results of our studies, we compare concentrations to water quality benchmarks. The benchmarks that we use are shown at the bottom. They can be EPA maximum contaminant levels, secondary maximum contaminant levels, health advisories, or health-based screening levels. A high concentration is defined as any concentration that is greater than one of those benchmarks. Moderate concentrations are concentrations between 50 and 100% of the benchmark for inorganic compounds, or between 10 and 100% of a benchmark for organic compounds. And low concentrations are less than 50% of the benchmark for inorganics and less than 10% of the benchmark for organics. For the data that we will be showing today, again, they are all from public supply wells, and we will not be including the secondary maximum contaminant levels. We will only be looking at MCLs, health advisories, or HBSLs. The distinction between moderate and low categories is mostly because of the fact that the inorganic constituents are present naturally in groundwater, whereas almost all of the organic constituents are present as a result of human activity and by default should not be present. Therefore, we use a slightly lower threshold for organics to be considered moderate concentrations. However, all concentrations have to exceed a benchmark to be considered high, and that is the focus of the presentation today. Let's take a look at the results of our studies nationally, starting with the inorganics. We evaluated 34 trace elements and major and minor ions, of which 19 have human health benchmarks. We also sampled for 8 radioactive constituents, of which 4 have human health benchmarks, and 5 nutrients, of which 2 have benchmarks. If we look at all 18 of the principal aquifers assessed, the results today show at least one contaminant at high concentrations in 14% of the 18 principal aquifers sampled to date, and moderate concentrations in 20%. If we look at the organics, we sampled 90 VOCs, of which 38 have human health benchmarks, 227 pesticide compounds, which includes both pesticides and many of their breakdown products, of which 119 have benchmarks. For the organics, low concentrations, again, are less than 10% of the benchmark. So if we look at all 18 of the principal aquifers, all 1,300 samples that we've collected in the last 6 years, we had no detections of organics at high concentrations, 2% of the detections at moderate concentrations, and 90% were at low concentrations. So there's a great deal of difference between the detection frequency at high concentrations for the inorganics versus the organics. Now we'll take a look at the specific contaminants that are prevalent at high concentrations. The highest frequency of exceedance was radium-226 plus 228, combined as the standard is set up. 5% of the samples we collected nationwide exceeded that benchmark. On the far right, you'll see a column that says the number of aquifers with exceedances. That's to give an idea of whether the exceedances are regional or spread out nationwide. Arsenic also at 5% exceeding the benchmark. Gross alpha activity, 4% exceeding the benchmark. Manganese is evaluated against the health-based screening level of 300 micrograms per liter at 4% nationwide. Strontium also HBSL at 3%. We evaluated radon using the alternative proposed maximum contaminant level of 4,000 picocuries per liter, and that was present at 2% using that benchmark. We did not evaluate radon at 300 picocuries per liter. The exceedance would have been much higher had we done that. Fluoride and gross beta activity come in at 1%. There are a few other contaminants that exceeded health benchmarks less than 1% of the time. Those included uranium, nitrate, molybdenum, lead, thallium, and beryllium. Now let's take a look at the geographic distribution of some of the contaminants that frequently exceed their respective benchmarks. First of all, we see radium, that's 5% nationally, exceeding the benchmark. But if we look at the distribution, you can see here the high concentrations in the yellow dots show the locations of where the concentration exceeded the benchmark, and they're clustered in the upper Midwest. That is the Cambrian Ordovician aquifer, where almost all of the exceedances of radon were found. There are a few in the Floridan aquifer, but generally speaking, if we look at radium 5% nationally, that's actually almost all due to the high levels of radium in the Cambrian Ordovician aquifer. A paper on the results of the radium sampling in the Cambrian Ordovician aquifer was recently published in the journal Applied Geochemistry. Arsenic is also high nationally, but it's a little bit more widely distributed. You can see there are some high concentrations in the West. However, pretty much all of the aquifers had several exceedances of the arsenic benchmark. Gross alpha activity is next. We see 4% nationally, exceeding the benchmark. And again, they are very clustered in the Cambrian Ordovician area, like radium was. Manganese has some regional issues as well, but more so in the glacial aquifers and in the Mississippi Valley and some up and down the East Coast. Radon, again, is a nationally significant at 2%, but almost all of the exceedances are in the crystalline bedrock aquifer of the Piedmont that you see there in the eastern part of the United States. Nitrate exceeded the benchmark only in 0.5% nationally. Those were scattered throughout the country in the high plains, the upper Midwest, and a few in the Valley and Ridge area of the country. In summary, we can look at locations where at least one contaminant of concern was detected at a level exceeding the human health benchmark in untreated groundwater. Although we don't sample for every single regulated contaminant, this provides some insight on the contaminants that are likely to be present in important principal aquifers of the United States. We also sampled for microbial indicators. This shows total coliform, E. coli, and enterococci. We did not have this data at every aquifer, so we're showing that separately. Of the 4 principal aquifers we see here, the Edwards Trinity is a karst aquifer. Glacial aquifers are much shallower than the others, and so you see higher frequency of detection of the microbial indicators when compared to the Colorado Plateaus and the Cambrian Ordovician aquifers. One unique finding was because we sampled for E. coli and enterococci in the same wells, although they co-occurred in some samples, 55% of the samples in which E. coli was detected did not have detections of enterococci, and 69% of the samples in which enterococci were detected did not have E. coli. These findings suggest to us that the E. coli and enterococci provide complementary rather than redundant information about the microbial quality of groundwater sources of drinking water and support the use of multiple indicators to assess the potential for fecal contamination. Some of the unique aspects of the NAQUA sampling are due to the fact that we sample for so many constituents. There are many emerging contaminants that we sampled for. Every one of the 1,300 samples that we collect, wells that we sampled, was sampled for more than 500 constituents. We have a full suite of radionuclides. What I did not show today was the results for lead-210 and polonium-210. We have a paper in production now on that topic. Many of the unregulated contaminant monitoring rule, or UCMR contaminants, are included in our data sets. We have a paper that is in process now on pharmaceuticals and hormones, which I did not show today. Also, a lot of the pesticide breakdown products and pesticides that are on the UCMR list are in this. And we have sampled all of our wells for the lead, polonium, pharmaceuticals, hormones, and the pesticide breakdown products. Very recently, we have added the perfluorinated compounds, and we are adding cyanotoxins to our sampling. We do not have data on those yet, but we are adding that to our sampling. In addition, we evaluate every single well for groundwater age dating tracers, and we work to extrapolate our findings to the full aquifer scale. I'll show you an example of that. This is an illustration of the screening level of age dating, indicating modern groundwater in the blue color, which would be recharged after 1963, pre-modern water in yellow, and a mix of old and young water in green. As you can see, the Biscayne in the southeast, Piedmont, crystalline, Valley and Ridge in the eastern area of the country, the glacial aquifers in the far north have predominantly young groundwater, and this makes them susceptible to anthropogenic contaminants. Where the other coastal aquifers, the NACP, that stands for the Northern Atlantic Coastal Plain, the coastal lowlands, south of Texas, Louisiana, and some of the western aquifers have a lot higher proportion of pre-modern groundwater. This makes those aquifers susceptible to a different suite of contaminants, such as those naturally occurring contaminants in those areas. I mentioned our efforts to extrapolate findings. The way we do that is to use machine learning methods in conjunction with our water quality data and explanatory variables. That allows us to provide what we call wall-to-wall and top-to-bottom coverage of aquifer conditions. In this example, from the Central Valley of California, we show the probability of dissolved oxygen being less than 0.5, less than 1, or less than 2 milligrams per liter. As you know, redox conditions are very important in controlling the presence of many other contaminants. And also on the bottom, we see the probability of manganese being greater than 50, 150, or 300 micrograms per liter. This was published also in a recent publication by Celia Rosecrans. So in conclusion, we asked the question at the beginning, what is the quality of groundwater used as a source of public supply in the United States? As I mentioned, we do not sample for every constituent that is regulated, but for those that we did, concentrations were less than the human health benchmarks in 84% of the groundwater from public supply wells. When there was an exceedance of a benchmark, radionuclides and trace elements are the contaminants that most frequently exceed human health benchmarks. That concludes my presentation, and I will address any questions during the Q&A session. Thank you.
Thank you very much, Bruce. And I want to let the audience know that you can go ahead and type questions at any time into the control panel on your, in the questions box in your control panel. And we're going to be holding questions until the end. So go ahead and type them in, and after all the presenters are done speaking, we'll go ahead and answer all of those. So next is Ed Furlong with USGS, and Susan Glassmeyer with EPA, and John Sullivan with Bellrica.
Thank you,
so
Deidre.
Are you ready to go, Ed?
Yes, if people can see my screen.
Yeah.
Okay.
Thank you.
Well, thank you, Deidre. I'd like to mention that this is a joint presentation and a joint collaboration between the USGS and the U.S. Environmental Protection Agency, the Office of Research and Development. At the USGS, this is specifically a project that has been a long-term collaboration from the Toxic Substances Hydrology Program, which is part of our environmental health mission area, but also collaborates extensively with and in our water mission area. I will be leading off with an overview of the studies. Susan will follow up with a couple of more interesting recent topical issues, and John Sullivan from Billerica will provide the perspective of a participant in this study. Our colleague Dana Colpin is also on the presentation, will help address questions, which we'll take at the end of the talk. So, without further ado, this was a two-phase study, started initially in 2007. We're going to focus on the second phase mostly today, to understand and assess the presence of contaminants of emerging concern, or CECs, in source and treated water across the United States. A national scale study, therefore. What we hope would come out of this, that would benchmark CEC occurrence and distribution in drinking water supplies and treated drinking water. It is a relatively limited number of sites, but we felt it was national in scale and covered a range of different treatment situations, as you'll see. The goal was to provide some exposure assessment for CECs and other legacy drinking water contaminants. There's not a lot of health benchmarks associated with many of these CECs, but we wanted to at least be able to provide information about exposure. And as I said, this is a joint collaboration of our two agencies. We've had a long record and very profitable record of production in this area. The results from this study have been, for the most part, published. One study on perfluorochemicals, or perfluoroalkyl substances, has now been submitted. But there are a series of papers, as you can see here. Those citations can be looked up in the recording of this presentation. But we have tried to make this as available as we can. And in this study, we looked at 247 compounds and element, 112 prescription, non-prescription pharmaceuticals and metabolites, hormones, 9 hormones, metallic, non-metallic trace elements, the per and polyfluoroalkyl substances, a number of other chemicals associated with inputs to source waters, including industrial chemicals, fragrances, number of viruses, bacteria, and fungi, protozoa were done as well. Just a real spectrum of chemical and microbial contaminants associated or potentially associated in source waters. An important aspect of the study that we really wanted to explain and elaborate today was that there was an extensive thought and effort put in to ensure that the results we produced were of the highest quality. We used paired, untreated, and treated water samples, which we collected taking the residence time of each plant into account. So we worked closely with each operator to ensure that when we collected an untreated and treated water sample, source water and treated, that we were, to our best of our ability, sampling the same parcel of water before and after it entered the plant. We only sampled once, so as I said earlier, this is kind of a snapshot in time, gives us a benchmark. We did, in the field, we collected 5 QC samples per location. That produces what you would call a 71% field QC sample load per site, quite a bit. We had a primary sample, we then had a replicate so we could understand our ambient precision. We also had a separate matrix spike sample that was collected both for the source and the treated water. At each site, we collected a field blank to help us assess any potential contamination from the sample collection process. In the laboratory, with every batch of 6 to 10 samples, we also had a laboratory blank and a laboratory fortified blank, or a lab spike. I mentioned below that 3 other things we did. I wanted to make sure I did not forget to mention them. That included a bioassay and then these other assessments that were based on a lot of this chemical data. And you can see in the lower right that sampling train approach. So there would be our source. We'd wait for the average residence time and then get to the treated side. In the next slide, I'll show you a bit about our distributions and demographic information. It's important to note that we kept the locations of participating drinking water treatment plants anonymous. That was a part of the initial study design. And what you see in the map to the upper left is the EPA regions and the number of plants that were addressed in each region, anywhere from 1 to 5 in each EPA region. So it is a national representation, but the total number of plants is a little sparse. The graph on the right shows you the population served by each type of plant as a bar graph. So we had 5 plants that served less than 50,000 individuals. We had, excuse me, 2 plants that's, or excuse me, 3 plants that served over 500 individuals. You can then see that we looked at a variety of treatment types, everywhere from chlorine, chloramine, to some combined oxidative processes. The majority of water source types was surface water, but groundwater sources were also included, and there were mixed source types. I mentioned all that QC. What did we do with it, and how did we use it? And this simple graph kind of illustrates how we tried to use the data, and the accompanying table will give you a little more information. If we look at all detections, we have both quantitative detections, that is compounds or elements that met all our qualitative identification criteria, but were also quantifiable, met our quality control requirements. They're there in that smaller blue circle. The larger gray blue circle represents the qualitative only detection, which we use for estimating frequency of detection. And in those cases, there were cases where they were below the LCMRL or reporting level, depending on what we had for each method, whether they're over 150% recovery, or if there was some positive blank detections, we might screen those. So when we talk about total detections, we're talking about the sum of quantitative and qualitative detections, but we will then break out the quantitative detections as a subset when we talk about concentrations. So how did we apply all those qualifying characteristics? So if, as we said earlier, if the concentration was less, as I said earlier, concentration was less than the LCMRL or RL, it was qualitative detection. And these columns to the right, you can see for source and treated sample, the number of affected measurements. A non-quantifiable detection, one where we could see the qualitative identification criteria were met, but we could not quantify it, those were qualitative. If we had matrix effects on quantitation, that is, recovery was greater than 150%, that was a qualitative detection. If sample concentrations did not exceed 3 times the field and or laboratory blank concentration, we would censor the result. And if our median recoveries were less than 50% for the laboratory 4 to 5 blank, the source or treated laboratory field matrix spike, we would censor analytes as well. And you can see there weren't that many. In sum, you can see that we qualified about 293 results in source water and about 170 in treated water. In total, we had over 4,600 to over 4,900 non-detects, and the quantified detections were on the order of 800 to 1,000 for treated and source waters, respectively. Important point to note, we spent a lot of time trying to make sure we had the most reliable data set possible, and in which case we found that the majority of compounds were not detected. But when we did detect things, this is how we looked at the data. So this is the total detections, the sum of qualitative plus quantitative detections. For pharmaceuticals as a class, perfluoroalkyl substance as a class, this represents over 120 compounds. Perfluoroalkyl substances, this represents 17 compounds, over 50 anthropogenic waste indicators. This is a variety of industrial compounds, fragrances, etc., and trace and major elements as inorganics here, and then microorganisms present in both source in the light green and treated in the blue bars. So you can see we've tried to organize these by treatment plant and ordinated it based on the pharmaceuticals frequency of detections. What's important to note that that things vary quite dramatically by plant within pharmaceuticals. Same with anthropogenic waste indicators and microorganisms, whereas for the perfluoroalkyl substances and for inorganics, there was a lot less variation. And Susan will talk about these a bit more, particularly the perfluoroalkyl substances further down. But this gives you a snapshot of what all the results look like for all the plants in our study. These are the compounds and elements that were most commonly detected. We saw at least 148 out of the 247 total compounds or elements being detected at least once in source water. And here you can see a list of compounds, the pharmaceuticals such as sulfamethoxazole, lithium, an element as well as a pharmaceutical, carbamazepine, an anti-epileptic drug, a number of other drugs that antivirals, diabetes control medications, stimulants like caffeine, just representing a real range of pharmaceuticals that are present entering source water. The perfluoroalkyl substances, these are all done by abbreviations. They're the sulfonates and acids. I won't go into a big breakdown, but they're very similar to many of the reports we've been seeing recently in news as a suite of compounds. The anthropogenic waste indicators, things like the antimicrobials triclocarban, triclosan, a range of other compounds, a full suite of inorganics, as well as a few microorganisms that we could see present quantitatively. When we went to treated water, we see things are much more reduced. And particularly wanted to note bromoform appears. Most of the other compounds have been eliminated, but bromoform was measured as one of these anthropogenic waste indicators, and it started showing up, not surprisingly. Again, a similar suite of inorganics. Now, if we look at concentrations by summed compounds and element concentrations by chemical class, we can see that in some pharmaceuticals were as high as 700 or 800 nanograms total in source water, and then dropping off dramatically. The perfluoroalkyl substances and concentrations, while they were very uniformly present when detected, were very different from site to site. Same thing with anthropogenic waste indicators and major elements and minor elements. We see more variability when we look at concentration. So there's more commonality between sites in terms of what's occurring, that is frequency of detection or number of detections for perfluoroalkyl substances and elements, but inorganic elements, but more variability when we start looking at absolute concentrations. Not too surprising. If we were to focus down a little bit on one particular class, and I chose pharmaceuticals because that's an area of my interest, we looked at 112 compounds. I apologize for the typo. 63 were never detected in source water, and 85 were never detected in treated water samples. So The majority were not detectable. The maximum number of quantified pharmaceutical detections in a given sample was as high as 20, and treated water is up to 5. The median numbers, however, were much lower, reflecting the fact that these compounds are relatively of low occurrence in source and in treated water, so being 2 and 1. If we look at the distribution of individual pharmaceuticals in all our source water samples, plotted for each compound as a box plot, the explanation is over here, just showing the box representing the 50th percentile at the hinge here, the top of the box, the 75th percentile, the bottom, the 25th, and then these whiskers at 10th and 90th, and then the mean plotted as this solid blue square. You can see that the range of occurrence is typically between somewhere around 1 to 100 nanograms per liter, low nanogram per liter concentrations for compounds like estrone. In the parentheses, we see the total number of detections, and by and large, only a few compounds are detected at any great frequency, things such as lithium, sulfamethoxazole, metoprolol, and carbamazepine. We can see as we move to the right that there are fewer and fewer detections where we're down to single detections. However, these concentrations that we see in source water are not too atypical from what we have previously seen in other surface water across the United States, but typically between 1 and 100 nanograms per liter, so subpart per billion concentrations typically in source water. We go to treated water, there's a slight shift downward. It's centering more around 10 nanograms per liter, and relatively few, or except for lithium, which is in micrograms per liter. I apologize for not mentioning that. But bupropion, metoprolol, some of these more persistent pharmaceuticals at low concentration, making it through a few of the treatment processes. Note that the number of detections are much lower, and there are much fewer compounds appearing. So it appears that overall treatment processes are removing pharmaceutical. So now I'm going to hand over the presentation to Susan so she continue on to focus specifically on peripheral chemicals and other topical aspects of our study. Susan?
Thanks, Ed, and good morning or good afternoon, everyone in the phone land. So yeah, as Ed mentioned, I'm going to talk a little bit about this current study on the fluorinated chemicals in drinking water, and then touched on 2 other aspects of our collaborative research together, which hopefully will be of interest. So the per and polyfluoro paper, this is part of the drinking water study. Unfortunately, it's a bit delayed from the rest of the study. We just submitted this, I think, 2 weeks ago, so hopefully by the end of the year, this should be finalized and pressed. But it's looking more at the detail that Ed had briefly mentioned on the PFAS substances in the source water and treated drinking water. So Ed, next slide. Okay, so this is a lot of information in one slide, but I'll talk you through it. The top graph and the middle graph are similar ones to what Ed just showed you when we talked about the rest of the compounds in the drinking water study. The top panel looks at the frequency of detection. The middle panel is the concentration. And on this, we did a log graph. So there is definitely still a disparity between the different concentrations, the different locations, but the log graph kind of brought everything up a bit. So as Ed mentioned, really the frequency of detection, specifically in the source water, was pretty constant between 10 to 14 of the PFAS 17 that we looked at throughout the study. But the concentration really varied by 3 orders of magnitude between the different locations. And if Ed can hit the little fun graphics, so we had utility 2 and utility 18. We saw a lot of significant differences between the source water and the treated drinking water when we looked at the Wilcoxon p-value, which is the non-parametric form of the student's t-test. And one of the things we think this is really linked to, and the literature bears this out, is the granular activated carbon that's used in these facilities. Both these changed or either recharged or changed their carbon fairly frequently and had deeper beds. So that could be one of the reasons why we saw a significant difference between the source water and the treated drinking water. Utility 24, it may look like there's a difference in concentration, but statistically there is no real difference between the source water and treated drinking water, despite the fact that there was some gap in this. It's just a longer time of treatment. So next slide, Ed. So one of the cool things about the study too is since we had the source water and the treated drinking water, we could look a little bit at fate transport in the source water. And it just so happened we had 3 utilities that were on what we call large river A, and we had 3 utilities that were on large river B. And you can see how very different the patterns are between the different PFAS analytes in there. So large river A was dominated by PFOA, whereas large river B was dominated by PFBA. And at the bottom graph, I just threw in the other utilities, just so you can see how the patterns were very different between the different utilities. Utility 4, that's one in the overall study, had the most analytes detected. Utility 22, for the PFAS compounds, they had the highest concentration. And utility 24, which is the groundwater location, just to kind of throw in a difference. So you can see, you know, how strong the transport and fate is of the different patterns along these rivers. Large River A, it was sampled over probably nearly a year, and the locations vary by maybe a couple hundred miles. Large River B, again, it was about a year difference in sampling times, and the river differences were over 1,000 miles between these different locations. So, Ed, next slide. So, to change things slightly differently, a couple years ago, in 2016, I think it was, 2015, 2016, Paul Westerhoff and Jocelyn Rice came out with a series of papers looking at their models that they did to estimate the factor water reuse in source water. So what they did was took a GIS map of wastewater outfalls and drinking water intakes and estimated flows, or the volume of water the wastewater put out, as well as the flows in the river connecting the wastewater outfall and drinking water intake. And with that, they're able to calculate a percent de facto water reuse across the country. So you can see in the map in the upper right-hand corner, those are the different communities they mapped out, and you can see some locations had, 10 to greater than 15% wastewater in their source water. And so this came out after we'd already collected the samples for the drinking water study. So I was really like, oh, why couldn't we have this data before we picked our locations, and then why couldn't we focus on these? But we had an opportunity then to collaborate with Paul and Jocelyn and another student in their group, Thuy Lin, to then look at our drinking water locations in relation to the drinks model data. So if we go to the next slide, this was actually published earlier this year in the Journal of the American Water Works Association. So the whole issue is on contaminants emerging concerns. If this is something that's interested you, I encourage you to go look up that issue. But again, a lot of data on one slide, so I'll try to talk you through it without having a pointer to point things at myself. The upper left-hand corner, Ed's going to access my Vanna White now. So the upper left-hand corner, we separate all the locations out by the stream order. So the 5th order streams are the smaller ones, the 9th order streams are the larger ones. So you just kind of separate them that way. And then within each order, we then organize it based on the percentage de facto water reuse, the median. So you can see our maximum locations had maybe 10 to 12% at median de facto reuse within each of the stream classifications. So then if we turn to the bar graph on the upper right-hand side, that is the number of organic chemical concentrations of the, sorry, let me back that. It's the organic pharmaceuticals and the anthropogenic waste indicators at each location. So just the number of locations, number of analytes. And the eighth order, the green bars, they kind of show the best where, you know, as the de facto reuse increases, the number of chemicals increase, which is good. I put my eyes, I can see it in the other one. Some people, you know, maybe a little more of an optimist than other people in the group, but I could definitely see it. But then when the bottom, we took the concentration summed for the pharmaceuticals and the antigenic waste indicators and plotted it against the de facto reuse percentage for each location. Utility 1 was a bit of an outlier. We had some blank issues there, which kind of threw out some of our concentrations. So it really was out. So if we just forget about that, you know, use the Heights rule of outliers that we can always throw out 1 point. Then if we look at the majority of the data, we ended up with an R square of 0.52, which means 50% of the variation between the data could be then attributed to the de facto reuse. So it makes sense, you know, kind of, yes, if there's wastewater input, you're going to see more CECs. So, but it's kind of nice to see the data actually supporting that, you know, gut kind of feeling that we had about the data. So if we go to the next slide. Okay, so, you know, we had the drinking water studies, but we really didn't know what the wastewater source upstream was. So we said, hey, let's on our next project work on that. So we went to one location that was a stream reach that had no tributaries coming in. So it's a really nice way to look at the wastewater and the drinking water. So with this, we had 6 sampling points we looked at. We went upstream of the wastewater treatment plant. We then had the effluent where the pipe was actually coming in the river. And you can see in the pictures on the right-hand side, that stone outcropping, that is where the wastewater effluent pipe is coming out. And you can see Jason in the middle picture actually sampling from the end of the pipe. You know, he gets the gold stars for actually sticking his hand in all that. So then we went downstream a bit from the wastewater treatment plant, which we call the effluent mixing zone, where the conductivity testing showed that the river was well mixed there. So we could look at the first step of dilution on the samples. Then we had a sampling point further downstream. Then we had the drinking water intake and then the treated drinking water. So ideally, we had tried to get these all residence time weighted, so we're looking at the same parcel of water. Real life did not really help. We had storms come up, we had some other issues happen, so we didn't always succeed on that, but we always did have this drinking water intake and the treated drinking water paired, so we had that worked out nicely. You can see from the diagram at the top two, we had grab samples at every location. Then we also deployed polar organic chemical integrated samplers at 4 of the locations in the upstream, the effluent mixing zone, the drinking water intake, and the treated drinking water. Today, I'm just going to touch on the grab samples. So I mentioned we had 1 stream reach. We did 3 sampling events. We did October of 2014, then April 2015. If you can see the picture at the bottom of Jason, he's actually standing on the wastewater outfall at that point, sampling down below. So you can kind of see the difference in the rivers between October and April. October and August look similar, that, you know, it was very easy to find the wastewater outfall. April, not so much. It's probably, the river was about 4 feet higher. And we've mentioned the 6 sampling points. So next slide. So again, a lot of information on one graph. So we have each of the 6 sampling points here, and then each of the pie charts. The dark blue, it's a compound was never detected there. The green was detected in 1 of the 3 sampling rounds. The gray was detected in 2 sampling rounds, and the dark green, it was detected in all 3 sampling rounds. And with this, you can really see how different the effluent samples were compared to those, you know, immediately upstream and downstream of them, and particularly then further downstream and the drinking water treatment plant. I should mention that we did the fluoridated chemicals we did not analyze in the 2nd round. So a lot of those were only detected twice. So that's part of the gray. You know, the green might have been a little bit bigger if we had sampled those in all 3 rounds. And additionally, the inorganic elements, we had a cooler get lost, so they were not sampled in the downstream. The drinking water taken, treated drinking water, or the first round. So again, that contributes a little bit to the size of the gray bar and the gray wedge and those 3 slots. But in general, the take-home from this slide is just wastewater effluent. Definitely we had more detections there and more frequency of detections. So next slide. Okay, so here we separated the organics from the inorganics, and as Ed had before, these represent both the qualitative and quantitative measurements. So we did the same level QC, everything was collected in triplicate. If something was below our detection limit, or if there was matrix enhancement present, we call that a qualitative detection. If things we were able to get a concentration on, that's a quantitative detection. But you can see really how different the organics and inorganics behaved. The organics tended to have more of a wastewater influence. So you see a higher effluent, the EFF samples, versus the inorganics tended to be more constant throughout the river reach, and that's the top right-hand bars. And then on the bottom, the left hand, we have the organic concentrations, and then the inorganic concentrations on the bottom right. And again, those are quantitative measurements. You can see, especially with the organics, how the April sampling, the green bars, were much lower than the October and the August sampling, the blue bar and the gray bar. So that's indicative again of the dilution effect we had from the higher concentrations. The effluent samples, they're the reason why the effluent, the gray bar, is lower than the blue bar in that sampling event is basically only due to sucralose. It had some matrix enhancement going on, so we had to make that a qualitative detection in the 3rd round, the August round. So that's it was literally like 20,000 nanograms per liter concentration, and so that's why that bar is so much smaller than the other one. So next slide. This is just looking at it for the compounds in particular, and you can really see the impact or not of the wastewater. So sucralose, as I mentioned, we didn't have the one measurement, but you can see definitely how the wastewater sample, the effluent, increases the concentration relative to the upstream, and then we have the dilution factor, the effluent mixing zone, then the downstream drinking water intake and treated drinking water. They converse that with the fluoride, with the upstream. That again, we had the wastewater input. So with the inorganics, we had some like fluoride that we could see this definite input from the wastewater. And for fluoride, it makes sense. That the community upstream is drinking fluorinated water. It's coming out in their wastewater, so they're going to get the increase there in the wastewater effluent. And again, so you can see the effect of the treatment in the drinking water treatment plant, where they're adding the fluorine or fluoride for dental health protection. So we saw the increase in concentration there. Maybe converse that with strontium, that did not see a really significant wastewater impact, and the concentration stayed fairly stable, particularly in the first round, the blue line, and the 3rd round, the gray line. The green lines, we had so much solution going on in the river system there. That's probably why it's appearing to tick up a little bit there, but definitely not much. But one of the interesting things was that the fluorinated chemicals, and here we just have PFAS as an example, one of them, they too did not show any strong wastewater correlation. That's more from whatever the upstream source is. There were some wastewater treatment plants upstream of our wastewater treatment plant, but again, it could be just runoff from locations. I need to look into more if there's any airports or any other types of known sources of fluorinated chemicals that could have been upstream of our sample reach. So next slide.
It's John's.
Oh, yes, all of your turn.
So John, you ready?
Yes, I'm ready.
Great.
Yes, I just wanted to talk about our role as participants in this enormous study from our perspective. There were negatives and positives, of course. The participation was an interruption to normal operations, but the overall study yielded important results. I remember distinctly poor Susan surrounded by 40, 50 coolers, doing all the logistics and everything, getting all the paperwork ready for overnight shipping and icing and all that stuff. And that's where she was working and where we get our chemicals. So all we could think about is, oh my gosh, are we going to get a chlorine delivery right in the middle of all this really, really, really important work she's doing. So that was kind of, nothing bad ever happened about it though. The commitment and time and effort and paperwork and sampling burden. We are kind of short-staffed here ordinarily, and even when we have an extra study like, say, the UCMR series, that is a burden just in and of itself. The scope of this study was really stretching us with respect to getting our regular work done and doing everything that Susan would have us do, like checking the coolers on delivery for breakage and contents and all that kind of stuff. So it impacted us kind of negatively that way. Positively, though, the study as a whole gave data on contaminants that we would not otherwise have collected. When people call us asking how our water quality is, we usually refer to the DEP contaminants that have, say, an MCL. So all of these other things that don't have MCLs attached to them, we don't know. We wouldn't know what to tell them. But because of joining in the study, we do have some data for them, and they've appreciated that with phone calls and emails. Collaboration and communication throughout the study. I'm an old school guy, and I grew up without emails and cell phones, and so to be able to get all of this work done definitely required all of that kind of stuff, and it was wonderful to see it all work. Added value to ongoing operations. The study for our community indicated that, if you saw from Susan's graphics, that our ozonation and frequent changeouts of our 4 feet bed of granular activated carbon seems to have helped with removing some of the source concentrations versus the treated water measurements. And also finding out that there's such things as the POSIS samplers. They're like a poor man's continuous monitoring instrument. It was wonderful to experience that. And we appreciated the organization of the sample collection logistics. As I mentioned, in the beginning, when we were asked to participate, we were thinking, oh, well, all right, maybe it's a great idea. But when the actual number of coolers were starting to arrive for both the intake and the treated water, we were right in the middle of a radio read meter change out operation. And there was a lot of competition for floor space during some of these projects. But overall, the experiences from the study that may be of interest to other drinking water treatment plants considering participating in a non-regulatory study, I would definitely recommend that people consider working with their regulators and don't be afraid of what may be found. In the very beginning, when we were approached by Susan if we would be agreeable to participating, the superintendent was concerned about what happens if bad things are found out. And we called DEP about that, and they said they couldn't really offer any kind of guidance about whether they would hold our feet to the fire if we did find out some bad news. But we decided to go ahead with it anyway, just on that, I don't know what you might call it, the human dimension to want to know what's out there so that maybe we can do something about it. And that was very worthwhile to us to be able to, we're actually glad we were asked as things ended up turning out. And so I would absolutely recommend to anybody that's asked to join in these studies, because it never stands still. There's always going to be new industrial contaminants out there that we don't know their concentrations until we actually go and sample. That's my takeaway today.
Thanks, John. So here are some of the take-home messages we'll go through real quickly. They are CECs are being increasingly observed in source and treated water, although more frequently observed because we're getting better at measuring these things at low concentrations. We would predict likely similar behaviors for nanomaterials or microplastic or other CECs yet to be measured, as John just alluded to. Things keep changing, coming along. We use complementary techniques that help us better understand the presence of these CECs mixtures and then proceed to assess their exposure, occurrence, and distribution, and ultimately assess potential human and ecological exposure. And having a cross-agency partnership, as we've had between EPA, between USGS, and with collaborators like John Sullivan, Billerica treatment plant, leverage the unique capabilities of all of us to fully address these drinking water questions and produce objective science-driven results. And our results have shown that de facto reuse of wastewater can result in these wastewater-derived chemicals occurring in both drinking water sources and ultimately in treated drinking water. So this is a wide range of collaborators who all participated, bunch of USGS, EPA folks. We also have academics participating and our great treatment plant operators like John. Typical caveat about this information is some of it's preliminary and subject to revision. We'd be happy to take questions, but would like you to hold them till the end so that Paul Bradley can speak on the next segment. So if Anthony, if you can switch sharing, that'd be great.
Yes, thank you very much for that presentation, and we will move on to Paul Bradley. And Paul, are you ready to go?
Yes, I am. Thank you, Deirdre.
Just a reminder, everybody, please type your questions into the question box and we'll answer them at the end. Thank you.
Thank you. So I'm Paul Bradley. I'm speaking on behalf of my USGS colleagues here that are listed on this front page. This research that we're talking about, we're moving towards the human exposure endpoint, so tap water research. And so this is increasingly a collaborative effort where we're working very closely with public health agencies and regulators, as well as the utilities. And arguably most importantly, we're now moving into people's homes. So the collaboration of the homeowners is very important to answering these kinds of questions. So as you saw from the previous talks,
Paul, this is Deirdre. We can't see your slides. I think we need to, if you could click on the button so we can see your screen.
I'll try it again.
Yeah.
Did it work that time?
Yeah.
Okay. All right. Thank you for that. So as you saw from the previous presentations, especially when we're dealing with surface water sources for our drinking water supplies, we're dealing with a reuse circumstance. And there's a wide range of contaminants that are being documented in these resources. And so a natural question that arises is, what's the potential for human exposure through drinking water, and by extension, what are the potential human health effects, if any, of exposures to these chemicals at the point of use? So in this case, tap water. So one of the questions, all right, so we've got a locked up screen.
So if you click on the arrow down at the bottom of your screen, the arrow pointing right, yep. There you go.
Yep, I got it. So one of the questions is, what kind of approach do you utilize? What kind of methodological approach do you take to have some sort of reasonable expectation of answering the question of exposure to tap water. And you saw from the previous studies that the chemical constituents in environmental samples can be quite complex. So we conducted a study in collaboration with the EPA back in 2012 to 14, where we looked at surface water, potential source waters under varying degrees of water reuse in 38 locations across the U.S., so a nationally distributed study. We used a combination of bioassays as well as extensive targeted chemical analyses to examine exposure. So in brief, the chemical analyses included analysis of more than 700 unique organic compounds. Among these unique organic compounds like pesticides and pharmaceuticals, which are specifically engineered to be bioactive and cause biological change at the molecular level, comprise more than half of the analytes. And then if we take a quick look here at the results of the study, looking at the upper left-hand corner, the red dots indicate the number of compounds detected in individual samples at these 38 sites across the nation. So the quick read of this is that the contaminant signatures in these potential drinking water sources can be quite complex, as you saw from this and the previous talks. To summarize it, of these greater than 700 organic compounds that were analyzed, we detected more than half of them at least once. And of the ones that we detected, more than half of those were pesticides and pharmaceuticals. Again, these are compounds designed to have biological effects at molecular levels. So we leveraged this experience and the experience that Ed and John and Susan just presented to you with respect to what's coming into drinking water treatment plants and what's potentially coming out of drinking water treatment plants. And the question then that we wanted to start addressing is, so what is the exposure at the endpoint where the customer is actually turning on the tap and getting the drinking water glass? And the first effort into beginning to answer this question was a pilot study of nationally distributed sites conducted in 2016. The analysis for this study is complete and the results have been written up. They are currently in submission to ES&T. So hopefully by the time we get to the December presentation, we'll have some results to show you. What I want to show you in this presentation is just very briefly give you some sense of the methodological approaches we're taking to assess tap water, some of the constraints we're working under, and give you an opportunity to give us feedback on this and future studies, and also to invite you to reach out to us if you want to collaborate, if you have specific questions that you think that a collaborative effort with us might help you resolve. So for this 2016 pilot study, the initial effort here is simply to look at a relatively limited number of samples, but distributed across the nation, just ask the question of what kinds of contaminants do we see in tap water. We looked specifically in half the locations were in the workplace, the other half of the locations were in home. For the home locations, half of those home locations were on public supply. The other half of the home locations were on private supply. So we're getting a kind of a home workplace comparison of drinking water and then a public versus private supply comparison of drinking water quality. For this study, we asked our research colleagues in the Toxic Substances Hydrology Program who were located across the country if they would collect samples for us at both of their workplace as well as at their home. A quick overview of the setup is samples were collected from 11 different states. We collected 26 total samples, drinking water samples, plus 2 blanks were collected during this study. Half of the samples from the home, 6 of the 13 were from a private well, 7 of 13 were from public supply. All of the workplace samples were from public supply, except for 1 sample in a workplace that had tap water as a drinking water source, but also had a commercial bottled water station as a drinking water source. So we went ahead and sampled the bottled water as a frame of reference in this study. So the sampling approach here, the basic question that we're trying to address is what's the exposure to humans? And so that informs how we're collecting our sample. So specifically, we want to know when a customer turns on their faucet, fills up their drinking water glass, what are they likely to run into? And to that extent, we are not policing the area. When we move into the kitchen, for example, to sample the kitchen tap, we are not cleaning it up. We're not removing the screens to collect the sample. If by chance the homeowner has a point of use treatment or an in-home treatment, we leave that in place. We just make a note of what the treatment is and the maintenance schedule and ask a few questions. And importantly, not so much for this pilot study, but for the next study that I described, we are not insisting on stagnant sampling. So basically, we want to know what the typical exposure is, and therefore the people collecting the samples for us were welcome to collect it whenever they wanted to. The analytical method, unique organics, again, preserving the pesticides and pharmaceuticals is important endpoints to analyze. The organic list is greater than 500 compounds. The inorganic list for this initial pilot study was 19 compounds. We're also analyzing microorganisms using selective plate counts to assess viability of potential pathogens. And then we're following that up with sequencing analysis to confirm the identity. In addition to that, we are using bioassay platforms, specifically binding assays for estrogen receptor, androgen receptor, and glucocorticoid receptor. So we conducted that study in 2016. As I said before, the results have been written up and are in submission now. The second study that we initiated was in 2017. And specifically, the question was, is within a specific large urban center, what's the variability in tap water exposures that we might run into? And so in this study, all of the source waters were public supply. And we collected samples at the tap water in the resident's home, but we also combined this with collections of raw water from the drinking water treatment plant, as well as treated water pre-distribution before it was released into the distribution system. Now, the sampling here between the raw water, the treated water, and the resident's home is not residence time weighted. So the raw water and treated water at the drinking water treatment plants is information provided as a frame of reference. So for this study, which was conducted in Chicago, the sampling approach is the same, but it's worth pointing out, specifically in the Chicago area, this is an old city with old infrastructure, and it has legacy lead pipe in the distribution system as well as in premise plumbing. So the specific note here that this study does not include stagnant sampling, so this cannot be interpreted as a worst case scenario for exposure to compounds that might be coming from the plumbing. The analytical method is similar to the pilot study in 2016, more than 500 unique organics. We increased the number of inorganics in this study to include compounds like arsenic, which were not included in the pilot study. And then on the bioeffects side, for the Chicago study, we are specifically increased our interaction with NIH, NIEHS. And so we're collecting samples and extracting those samples and then running them through NIH's high throughput bioassay robot setup to directly assess the potential biological effects of those tap water samples. So the Chicago study, we collected 15 tap water samples from each of 3 different service areas for 3 different drinking water treatment plants. 2 of these were in the Chicago area, the Jardine and the Sawyer drinking water treatment plant, and then there were 2, at the time of our sample collection, there were still 2 facilities in operation in East Chicago. And this is just a summary slide to kind of give you an indication of the level of, from the comments that you heard in the previous talk, the level of logistical challenge associated with moving into a home and collecting these samples. We're collecting roughly 30 bottles for each sample. So on that picture on the lower right-hand side, that cart represents 8 samples collected during that day. So you can see from all 3 of the talks presented here today that the USGS and the EPA and other federal agencies are conducting research on our drinking water resources, from both the ambient supplies up to now with this most recent work to the potential human health exposure point at the tap in people's residences. And so this, as I said at the outset, this is a highly collaborative effort, working very closely with public health researchers and with the public and with the utilities to kind of begin to understand the level of concern that's appropriate. And that's it for this talk.
Great. Thank you very much, Paul. And I just want to remind everyone to please type your questions into the questions box. And we do have a few questions already. I think the first ones are primarily for Bruce, who was talking about some of your groundwater studies. And so the first question is, do you have an estimate of PFAS occurrence in groundwater sources of drinking water? I know you had said you don't have the report available yet, if I remember correctly, but maybe do you have some preliminary analysis you could share with us?
Not from this program. We just added the perfluorinated compounds this year, so the first round of data is just coming back. There are other USGS researchers who have done some in other geographic locations. Most of them are kind of local scale, but at the scale that we are sampling, this was the first year we collected the data, so we don't have that available.
Okay, thank you. And then can you also explain, Bruce, the use, the sampling that you did in private wells versus the public water system wells?
Yes, the data that I showed today was all from the public supply wells that we have sampled. The NAQUA program started in the 1990s, and we had a design that was based on what we called land use study networks and major aquifer studies. A land use study network has a very small, relatively small footprint within an aquifer, typically monitoring wells that represent one specific land use, usually urban or agricultural land use. The major aquifer studies are a larger part of the aquifer, but not the entire principal aquifer. And they're homeowner wells that we have sampled and, again, at the depth used for domestic supply. Those networks that were sampled in the 90s have become part of what we call our decadal trends network. A subset of those samples were, of those networks, we have about 80 networks of about 30 wells each that have been sampled in the 90s, again in the early 2000s, and some of them now for the third time. So we are using that data for both status evaluations and for trend analysis. So when we do that three-dimensional extrapolation, we use our data from the public supply wells, the domestic supply wells, and the land use monitoring well depth. And then we also use those two smaller scale studies or those network types for our evaluation of changes in concentrations over time. And there's a website that has that all on it. If you just look for groundwater quality trends, you'll find a website that displays the results of that.
So I think you explained this a little bit, but the difference between the use of the shallower wells and then the wells with that are much deeper. So I think the question was, why does your study exclude the shallow and moderate depth public water supply wells?
The overall study, so we were just addressing the public supply well issue today.
Right.
The overall study is addressing the question of groundwater quality in general, and that overall study does use the monitoring well networks, the domestic supply well networks, and the public supply well networks. So what we discussed today was one part of a larger question. The question we addressed today was water quality in public supply wells, but the overall question is groundwater quality. And when we go to answer that question, we use all three networks. Does that better address the question?
Yes, I think so. Thank you very much. And then just in terms of the maps that you showed with the contaminant occurrence throughout the U.S. with the dots on it, are the areas on the maps, can you explain if there's no data there or if it's just because there's no major aquifer there?
The
Go ahead.
The shaded areas on the map are the principal aquifer locations that we sampled. There are other principal aquifers that we did not sample, and then there are other places that are white space because it's deemed other aquifer type that is not a principal aquifer. So there could be, what I displayed is what we studied in this last 5-year period. There are other NAQUA studies of those shallower monitoring and domestic supply well networks that would fill in that map, but not at the depth zone used for public supply. Does that answer that question?
Yes, I think so. And there was another question about the availability of the NAQUA study, and I think that the question was referring to the principal aquifer study, which is available on your NAQUA website, if I remember correctly.
Yes, we publish, for every year of study, we publish a data release that has the actual data, and then we also do a fact sheet that summarizes the results for that particular principal aquifer. We have, I believe, 11 of the 18 published already, and we are working on publishing. There's a delay. I displayed, the reason that the provisional subject to revision was on the slides today is because I included all 18 of the studies. That includes the most recent, but I believe we have 11 of them published. So you can find the fact sheet for each of the principal aquifers that has been completed and a data series report for each of those that are accessible.
Okay, great, thank you. And I'm not seeing any other questions, so please type in your questions if you have them. But I do have a question for Ed and Susan, myself, and just about the study where you were looking at the PFAS concentrations. I was wondering if some of those pilot utilities had also done sampling under UCMR 3, and if you're finding some different results, or if you're going to be comparing the pilot results to the USGMR 3 data results for PFAS specifically, or even some of the other contaminants.
Susan, do you want to address that? Or
Sure, I can do that. So, yeah, of the 25 utilities, I believe I was able to find the data for 23 of the 25 in the UCMR database. Our detection limit was much lower than that in the UCMR. Most of ours were at 1 nanogram per liter or less detection limit, whereas UCMR compounds were somewhere between 10 and 40 nanograms per liter, I think, for the detection limit. So, as a result of that, we had a much higher frequency detection in our study than what they did in the UCMR. We only had one location that exceeded the new health advisory guidelines, the 70 nanograms per liter, PFOS and PFOA summed. That was the utility 22. And that was one of the reasons why I wanted to go look and see the UCMR data, just to see if that utility was in an exceedance for the, during the UCMR. They were not. There were no, I believe there were no measurements made. So, sometime in the interim between our sampling events, which was 2010 to 2012, and UCMR, which is 2013 to 2015. Something changed either in the source water or the treatment or something. So there was definitely a difference in that one utility. So, you know, their consumers are a little more protected now. Did I answer all your sub-questions about that? Or?
Yes, yeah. And then just if you could tell us again when that study is going to be available. I can't remember if you said the end of the year.
It's submitted now, so just as quickly as people are reviewing it right now, and once we get those, we'll turn it around. So hopefully by the end of the year, it'll be, you know, at least available on the web, if not, you know, finalized. But right now, it's out of our hands.
Yeah.
Okay, great. Thank you. Okay, and I'm not seeing any other questions coming in, so I just want to give a big thank you to all of our speakers today. I think your presentations were very informative. It's always great to hear about the work that you're doing, and I think it's very valuable for ASWIS members and others to hear about it. So we really appreciate that, and we appreciate the whole audience for listening in today. As I mentioned previously, we will have a recording of the webinar available on our website, and so you can go look at the recording and also the presentation slides on our website, along with the previous webinar recordings that we did with USGS back in April and July, and many other topics as well. So with that, I think we'll go ahead and end the webinar.